Welcome to Session 3 of Week 7 in Virology Part 1. This week we're talking about DNA replication. And in this session, I'd like to talk about how DNA can be primed via DNA or a protein. In the previous sessions, we talked about DNA synthesis primed by a short RNA fragment. Today we're going to use either DNA itself or a protein as the primer. And this is important because, as you know, DNA synthesis is always primer dependent. Let's first look at the Parvo viruses. These are viruses with single-stranded DNA genomes. There are about 4,000 to 6,000 base pairs in length. Here's the schematic of the genome at the top here. And this genome is largely single-stranded. As you can see, it encodes just 2 open reading frames, 2 proteins. The ends of this genome are quite unusual. They consist of terminal repeats, that is, the sequence is the same at each end. It's repeated at the genome termini. And not only is it repeated, but it has unusual properties that allows it to form this T structure at either end. And the reason why it can form this kind of a T structure is because the sequences in these end regions, these termini, have inverted complementarity, so they can base pair with each other. So you can see here is the sequence of this T structure here at the left end of the genome. And you can see there's a free 3' hydroxyl here, which is the end of the 3' end of the viral DNA. And this can base pair, the blue part shows you where it can base pair with another region of the viral genome not too far away from it. So that's one region of double-stranded DNA made by this sort of internal base pairing. And then at the very left here, you can see another kind of double-stranded DNA structure, again made by base pairing within the viral DNA. So that's why there is a T structure at either end, because you have this internal complementarity. This turns out to be the primer for viral DNA synthesis. When you think about it, it's perfect. It's a 3' hydroxyl, and there is DNA upstream. Actually, all you need is a 3' hydroxyl, and the way that's provided, of course, is by a primer. In this case, it's the viral DNA folding back on itself. So the polymerase is going to initiate replication right here and start adding bases complementary to the template, which is the single-stranded DNA. So let's see how that works. So we start at the upper left, which is a linear version of the parvovirus genome. Now it's shown here as a single line. There's no secondary structure drawn yet because I want to show you these terminal repeats. So remember, these are the repeated sequences at each end of the genome. What I didn't tell you is that they're inverted and they're complementary. So that's why it's ITR, inverted terminal repeat. Here, the repeat is just diagrammed in a schematic way as A, B, C, A prime, D. At the other end, it's repeated, but it's inverted. A prime, B prime, C prime, A, D prime. The prime means the complement of the other. So here, the A is the complement of A prime. So these would base pair if they were near each other. And that's shown here. So this genome can form this bottle-like structure, if you will, where the 2 inverted terminal repeats hybridize. So this could be an A or a T, a G, C, A, T, T, A, C, G. The 2 ends are not only inverted and repeated, but they're complementary, so they can base pair. So this genome can adopt a variety of interesting structures via these inverted terminal complementary repeats. They can form this bottle, but this is not very useful for viral DNA synthesis because there is a free 3' end, but there's no template to copy. The 2 possible structures that are useful for replicating DNA are either this one on the right, so here's a 3' hydroxyl from this T structure. That could be a primer for DNA synthesis. But let's make it simple to look at and consider this structure on the left, where each end of the genome has base paired to form a T-like structure separately. So the polymerase will take this 3' hydroxyl as a priming site. And this, of course, is the cellular polymerase, and it recognizes this as a DNA that can be copied. Now, interestingly, this doesn't require T antigen, as does SV40. This is replicated by the cellular polymerase as it is. And so the polymerase elongates on this 3' end and makes a copy of the template, which is in blue, and the product is in red here. The polymerase extends all the way to the end, and we've melted out the T structure at the right-hand end so you can see how the polymerase makes a copy all the way to the end of the genome RNA. Okay, now, the problem here, of course, is that even though we have a primer, we're still missing some sequences in the genome. So you can see the red doesn't cover the entire left-hand part of the genome. So how do we do that? Well, remember I told you that every DNA virus needs at least one protein to be made, one viral protein to be made in order to replicate its genome. And for Parvo viruses, that protein is called Rep78/68. What this protein does is it nicks the DNA, in this case this double-stranded DNA, between the A' and the D segments and makes a free end, a free 3' end. That serves as a primer for the DNA polymerase. The DNA polymerase then proceeds to copy this strand and completes the copying of the genome. Now we have a full-length double-stranded copy of the viral DNA. This version up here was not. It was missing left-hand sequences because of the folding of the primer. But now because of the nick, so this nick is really important, the nick allows the polymerase to copy the left hand of the genome. So there's no end problem because of the nicking. That gets rid of the end problem. It gives you a primer to fill in the left-hand missing sequences. So now we have a double-stranded product. What happens next is the polymerase takes these ends on the left, can fold into their interesting T structures. The polymerase can initiate on this 3' hydroxyl and start to copy this strand again. This is strand displacement now. You can see the synthesis of one strand is displacing that of the other. You now have a displaced strand, this one here in number 7, which is basically another piece of genomic DNA. You've now replicated the genome, and now we have a new double-stranded product which can go through the same nicking and extension reactions as before. So these simply go through this series over and over to make more genomic DNA. So this is continuous replication. You don't need any discontinuous replication. It's all continuous 5' to 3' in one direction. We don't need polymerase alpha to make primers. You don't need a primase. It uses the inverted terminal repeat right here to self-prime. All it needs is polymerase delta, RFC, and pCNA to make that sliding clamp. And this Rep78 is an incredibly important viral protein, right? It makes the nick that allows you to finish copying the genome, and it has a variety of activities that are involved in that. And again, there is no replication fork in the replication of this genome. It's all strand displacement. So that's a neat solution to the primer, the requirement for a primer, and by doing that, there is no end problem. Pretty neat. So that was priming with an unusual structure in the DNA. Let's look at another solution to getting a primer, and this is done in adenovirus-infected cells, and this is to use a protein. Let's first look at the genome of adenovirus. Now, these are viruses that are slightly bigger, substantially bigger than the Parvo viruses we've just looked at. These are double-stranded DNA genomes. Okay, so they are double-stranded, 2 strands of DNA, different from the Parvos. And they have free 5' and 3' ends. They're linear, double-stranded DNA molecules. There is an origin of replication at each end. Now let's go back and point out that the origin of parvovirus replication would be right here, this 3' hydroxyl, and the region in that area is the origin of replication for that virus. Adenovirus has 2 origins of replication, one at the left end and one at the right end. You can see they're labeled here ORI. And interestingly, there is a protein linked to the 5' end of each strand. It's the little orange ball here. Here's one linked to the bottom strand, 5' end, and here is one linked to the top strand, the 5' end. The replication of this genome is by strand displacement, as you will see. Of course, it is semi-conservative, as for all the DNA replication that we're talking about. So let's see how this works, protein priming. Let's start at the upper left here. We have an enlarged view of the left end of the viral genome. Remember, it's a double-stranded viral DNA. And at the very left end, we have, of course, a 3' hydroxyl on the bottom strand. This virus, adenovirus, encodes its own DNA polymerase. And that DNA polymerase is shown here as a purple sphere. And this DNA polymerase binds to a second viral protein called PTP, which stands for pre-terminal protein. This is a precursor version, if you will, of the protein that ends up being linked to the 5' ends of the viral RNA. The DNA polymerase binds PTP, and it also binds the 3' end of the genome, the origin of replication. And then what it does is the polymerase adds a C residue, one of the 4 bases, C, from a precursor of CTP. It adds it to a serine. It covalently links it to a serine of the pre-terminal protein. This complex then serves as a primer for viral DNA synthesis. So remember, the function of a primer is simply to give you a 3' end on which to add more bases. And that 3' end is typically provided by another base. In this case, there is a single base that supplies the 3' hydroxyl. It's a C. It happens to be complementary to the first base at the 3' end of this strand of the DNA, and it's linked to this protein to which the polymerase is attached, and the whole complex sits at the 3' end of the genome. So that's the primer for viral DNA synthesis. A protein stuck on the polymerase then binds to the 3' end of the DNA. That C then serves as the primer for new DNA synthesis. So the next base that's going to be added by this polymerase would be an A, complementary to this T, and it'll be covalently linked by a phosphodiester bond to this C. So the polymerase is shown in here in this diagram now, starting to move along the template strand. This is displacement synthesis. It's displacing the blue strand here. It's moving down the genome. It's making a complementary strand, which is shown in red here. So now the pre-terminal protein is left behind because it is linked covalently to that first base, the C, and the polymerase is now making new DNA. And as it moves down the viral template, it displaces the other strand. That's called the displaced single-strand template DNA. And as that single strand is displaced, it binds to another viral protein called the DNA binding protein, or DBP. That's the yellow sphere shown here. And these bind along the length of the displaced strand, and they keep it single-stranded. They keep it from hybridizing to form a double-stranded DNA. And eventually, the polymerase makes a complete copy of the template strand. That is shown here. It's now a double-stranded product. So you have the original light blue strand. You have the newly synthesized red strand, which of course is linked to a primer, and the blue strand has a primer from the previous round of synthesis. So this is a new genome. The terminal protein is then processed. The pre-terminal protein is then processed by a protease to give you the final terminal protein, and that's shown in orange here on the genome. So the pre-terminal protein is pinkish, and the terminal protein is orange. The other strand, what happens to that? The one single displaced template strand coated with DNA binding protein. That's shown here. Now, the ends of adenovirus DNA happen also to be inverted terminal repeats like the ends of parvovirus DNA genomes. And so they can hybridize, as you can see here. They're complementary. So they form this structure, a kind of a panhandle structure, I guess, if you will. Here's the 5' end of the viral DNA bound to terminal protein. The rest of the genome is coated with single-stranded DNA binding protein. And then you have more base pairing. And what do you think? That can serve as a primer for DNA synthesis. So the polymerase will come on to the 3' end right here. This structure, this double-stranded structure, looks very much like one origin of viral DNA. And in fact, it is an origin of DNA synthesis. It's the other end right here. And the polymerase comes on with another molecule of PTP linked to C and initiates replication at the 3' end. So here the protein is linked to the 5' end, so that tells you that this is the 3' end. This will serve as a template now for synthesis of DNA, and that's what's happening in this picture. The polymerase has initiated synthesis with pre-terminal protein right down here, and it is making a complementary strand. And in so doing, it's making it double-stranded and pushing off the single-stranded DNA binding protein. So the synthesis initiates right here at the 3' end and goes in this direction, copies in a 3 to 5' direction, and synthesizes in a 5 to 3' direction. So that gives you another double-stranded product. And that's why, of course, it's semi-conservative, because both strands are replicated. And the key here, of course, is a protein primer that not only serves as a 3' hydroxyl for the addition of new bases, but doesn't give you any empty sequence at the ends. There's no end problem with this virus either by use of this protein primer, because the primer is right here at the last base of the DNA genome. Nothing has to be removed. No RNA primer has to be removed. So that's a really cool solution to priming and the end problem. This is just a look at this adenoviral single-stranded DNA binding protein and how it works as the polymerase is synthesizing new DNA. That's shown in red on the blue template. The DNA moves along, and here we have strand displacement. You are now denaturing the double-stranded DNA, and you have a single strand being displaced. In order to keep that single-stranded, it is bound up by sequential copies of this single-stranded DNA binding protein. And that keeps the genome single-stranded until such time as this synthesis is incomplete, and then you can form the panhandle structure and go through another round of copying.